only the sequence data you have, you just tell the software to develop a phylogram, it will develop a phylogram, but you cannot tell or you cannot infer anything from that data, because you do not have any time concept in that. But if you have time, also put it in, means your data is also related to the time. For example, you are analyzing some microorganism from some fossil, something like that, and you know that carbon dating this fossil is from this age, then you can do analysis, and then you can tell that how that evolution takes place in that particular organism and other things, then you can, that is justified to use the phylogram. But if you do not have that thing, do not do phylogram for the sake of beautification. So, cladogram, the same thing here, time concept is not there. As the time concept is not there, so all these things are in one line. So, all the sequence, if you see, they will be just staggered at one point and all the branch is there, because there is no concept of time here. So, this is called a rectangular cladogram or phenogram. There is another which is called slanted cladogram. Okay. There are two types of cladogram, rectangular cladogram and slanted cladogram. Again, what for these two? Many students use this kind of things without knowing what for this is required. So, rectangular cladogram is required when we want to do grouping for the taxonomic studies. Just to group for the taxonomic study, then only use this rectangular cladogram, do not use slanted cladogram. Slanted cladogram again is used to do the evolutionary studies. It is not related to the time, but in the evolutionary study there are many aspects. For example, ape and human started their journey from one point, then they separated out. So, they diverged out. Okay. Human and rice also originate, but they originate independently and they are growing. So, this kind of evolutionary thing is called how the origin evolves. One is divergent origin, one is parallel origin. The two organisms develop parallelly. So, they are called parallelism. Two organisms once upon a time they were same, then diverged out. So, divergence. It may be possible two organisms due to the mutation and mutation they may converge also in future of time. So, to understand this kind of things, convergence, divergence and parallelism, we need to use this slanted cladogram, not this. But if it is only taxonomic grouping, most of us mainly do this, only try to restrict rectangular cladogram, not the slanted cladogram. Slanted cladogram means when you want to know convergence, divergence or parallelism in the evolutionary study, then you use that thing, because this is kind of convergence, that in future these two sequences, though they are originally from two, but due to the continuous mutation in future they may converge and again gives one single entity. This is called parallelism, parallelly they are evolving, and this is the divergence, that it is started and then diverge, again going diverge. So, it is divergence. So, if you want to explain your data with this kind of things, then use this. If you don't want to explain your data with this point, what for you are using then? Okay. So, based on your interpretation, your data, you have to choose what tree you will put it in your thesis or result or anything. Okay. So, do not take for beautification purpose, only for the purpose that whether you can explain your result, support your result with this type of tree, then only use that thing, otherwise whatever purpose you use for that purpose.
The two parallel lines that you are talking about, in the same parallel line when you have three components, that is also divergence.
So, that is diverge from here. So, in this process of evolution they are diverging, but they are independently, they are parallelly evolving. They are parallelly evolving from one thing.
So, the situation is not mutually exclusive also. Please remember that when we are having this kind of a data, the same data when you start at the same time, parallelly two components evolve, but at some point of time they also have three different divergences also.
Three different divergences, but they are also evolving parallelly.
Sir, in parallelism, what is the similarity between these two parallel evolving organisms? What will be the similarity?
That is this parallel evolution, actually the means that is why the lineage transfer of the gene takes place, because it is everything is there in 1, then it comes out from due to some mutation, it comes out from 1. So, the genes which they have, they also carrying that thing, again after few million years they also evolve like that. So, in that way what happens, they carry the same linear genes and they are present in different axes. Okay. So, yes.
In this case where there are three, firstly they are diverging and then they are moving parallelly.
Yes.
So if the three parallel lines are closer
Hmm.
or they are more far, so what is this going to indicate?
This length is not having that means this length, that has no, because that you can in the software you can increase that thing, only the length, not the height. Height you can change, height has no meaning on this, only the length has the meaning, not the height. So, either this height or this height has not that meaning, it is the meaning is these lines, the parallel means horizontal line, not the vertical line. Okay. So, the tree can be presented as rooted dendrogram or unrooted dendrogram. Rooted dendrogram means the evolution started from one particular parent and then they started evolving, and this is unrooted dendrogram where there is no root. If you want to study the evolutionary relationship between human, rice, mosquito, tiger, so you cannot think that they may have one ancestor. Okay. But if you want to understand the evolution of different bigomoviruses, then all the bigomoviruses must have coming from one bigomovirus. So, in that case you have a rooted dendrogram. So, again depending upon your data set, if your data set do not have a probable means ancestor parent, you use this, but if it is having, you do not have to use this. Many students goal map, this goal map means that is unrooted. You are trying to do a Magnaporthi sequence analysis, goal. Why? All the Magnaporthi different isolate may coming from one particular ancestor. Okay. Then why goal? That means they do not have any common ancestor. But definitely if you want to do Magnaporthi and other different fungus, then also this kind of things is okay. But if one organism, one particular thing, different isolates you are using, definitely you have to use rooted tree.
One genus, many isolates or strains of
Okay.
that genus.
So, why I am trying to tell, because software you click that kind of things rooted, it will give root. If it is unrooted, software will not dictate you, you have to dictate the software what you want. So, assume that all taxa derived from a common ancestor and assume that all taxa do not have a common ancestor. Then there are two different methods of generating this phylogenetic tree. One is called character based method, another one is called distance based method. Character based method means, suppose these two are the sequence, we are taking individual nucleotide wise means analysis, this T is mutated to G. So, it will generate a score value and then it will generate a matrix and then it will generate a tree. But the distance based matrix means, suppose these two sequences are there, one is T, one is G, another sequence you have added, here also T. That does not mean that this sequence is from here, it may be possible that T is there because the original thing is there, then it is score 0, but it may be possible that one generation it was G, then next generation again it revert back the mutation to go back to this. So, due to this generation process, there is a score is 0, but the genetical distance is 1. Okay. So, that may be possible. So, different software can analyze that kind of distance based approach and then they will generate again a distance matrix and then they generate tree. So, there are character based approach and distance based approach. So, these are important character based softwares or algorithms which we use: maximum parsimony, maximum likelihood. When we use maximum parsimony, again software will not tell you what you have to choose. In the software you will have maximum parsimony approach, maximum likelihood approach, you have to tell the software which you want. Now, if your sequences are quite similar, different strains or isolate of a particular pathogen, and if you have relatively less number of sequence, then you use maximum parsimony method. If your sequence is much more diverse, and much more intense, then you do the maximum likelihood method. Okay. So, if you are trying to analyze, for example, in our case I want to analyze a potyvirus, a bigomovirus, another virus. So, I will use maximum likelihood and maximum parsimony when I will use the different isolates of bigomovirus. Okay. So, when it is very much close, because close means almost all the nucleotides will be same, only few nucleotides will be different. Clear? So, character based approach is perfect for that. Maximum likelihood means maximum nucleotides are different, only few nucleotides will be matching. So, again the character based approach is there. Okay. But if it is in between, that some of the sequence stretch is matching, some of the stretch is not matching, then we have to use distance based method, and that is I am coming. So, this character based method, just one example how the software does actually. For example, I want to analyze human, rabbit and chicken, this thing. So, 1 mutation, 2 mutation, 3 like that. So, it generates a score between human and rabbit 1, between human and chicken 3, and between rabbit and chicken again 3. 3 mutation, 3 score. Very easily I am telling, it is not like that, software tells so many things, but for your understanding very easily I am telling. So, if that way score will create. So, if the score is creating, then how the tree is developing, that means this human and rabbit our score is 1, that means the least mutation is taking place. So, that means they are much more closely linked. If much more mutation is there in this case, that means they are much more distant. If less mutation is there, they are much more close. Maximum parsimony, they are maximally parsimonious, close together. So, human and rabbit are close. So, you take human and rabbit 0.5, 0.5. So, this is the 1. So, you divide that thing and then you join with the chicken. So, this is 1.5 and 1 and this 0.5, that means 1.5 and 1.5 3. Human and chicken is 3, then rabbit again 0.5, 1 and 1.5, again rabbit and chicken 3. Okay. So, in that way they generate the character based approach generates the tree. For the distance based approach, the example is neighborhood joining approach. So, how, for example, I want to study spinach, rice, mosquito, monkey, human, different things. Okay. So, a distance matrix will be created by the software, and we will now find out which one is mostly related. For example, here the distance is very less, 3.3, that means these two, monkey and human, they are closely related. So, first step we make a group monkey human, make a group monkey human. The second step we have to understand with this monkey human group which one is closely related. So, again the software will generate another table next cycle. So, now it will take monkey human as a group and try to analyze with this sequence, and then with this group they try to find out, and they observe that with monkey human mosquito is coming closer. So, now they join with the mosquito. So, the branch is growing, and then mosquito monkey human they will find is as a group, and then they will try to analyze with what they are more. Now, another cycle, but here we observe that it is not between monkey human, the close relationship is between rice and spinach. That means, there is another evolutionary group. So, this is called polythetic origin of group. So, this is one origin, this is another origin. So, spinach rice will be one origin, this will be another origin. Now, they will put together to form another the super group. So, this is one clade which is having this is second clade, and these are parallel evolution, and this is divergent evolution like that. So, you can now explain. So, how the software actually does this analysis and generates the tree. Clear? You do not have to do, you just have to click, but you have to choose whether you need a distance based method, whether you need a character based method, that you have to choose, but software will generate the result for you, and you have to now infer that what actually you want to say. The last thing is that you have to do bootstrapping. Bootstrapping means judging the confidence. Suppose if you have a group of people and I told you, you make your partner pair, you make your pair what is ever available, and for example, some boy is pair with one girl, now another girl will come, he will leave that girl, join with another girl. Okay. So, bootstrap will actually tell how much is really they are having the chemistry to stay together, and if they do not have the high bootstrapping, that means if any another girl will come, that boy will go. Okay. So, bootstrap will tell that okay, I have today I have taken 10 sequence to do a phylogenetic analysis to make a grouping. If tomorrow 11th sequence will come, whether that 11th sequence will disturb this harmony or not. Okay. So, this bootstrap value will is a method to assessing the confidence of that grouping. Okay. If the grouping confidence is very low, that means these two sequences today may be staying together, but if any new sequence will come, they may separate out and they may form another group. Okay. So, they will judge these things by doing the continuous bootstrapping, means 10 times or 100 times or 1000 times they will analyze the sequence, taking out one sequence. Suppose you have given 10 sequence, they will analyze one sequence with 9, then they remove one sequence, then with 9 sequence they again do the analysis, and in that way they do bootstrapping. जैसे bootstrap करते हैं, so कितना बार कैसे करोगे और हर बार एक ही टाइप का हो रहा है क्या नहीं, and then they will tell that whether the two pair which are coming together, how much after 100 times or 1000 times if you do, how many times is the probability that they will come together. So, the value will be 90, or 90 means in 100 times 90 times they are coming together. If it is 10, that means in 100 times only 10 times they may come together. So, there may be possibility if any other partner will come, that group will break. Okay. So, final, there should not be any confusion. So, if you have a set of sequence data, then what you have to do? You have to obtain a multiple alignment using any software, and after obtaining multiple alignment, you have to ask the question that whether there is a strong sequence similarity or not. If there is a strong sequence similarity, then if it is yes, then you do maximum parsimony method, because strong sequence similarity means they are close very. If they are close, only 1 2 nucleotide difference, you have to do the maximum parsimony method, and then do bootstrapping and generate the tree. If it is not, if the sequence similarity is not strong, then the question is there is clearly sequence similarity? The sequence similarity is clear or it is totally different? If the question is yes, it is clear 60% or 50% or 70% similarity, not the 95% similarity, but 60-70% similarity is there, then go for distance based method, that is the neighborhood joining method. Then again do the bootstrapping and generate the tree. And if there is not at all strong similarity, only 20%, 30% similarity, so much more sequence is diverse, only few sequences similar, again it is character based approach, do a maximum likelihood method and generates the bootstrap and generates the tree. Clear? So, depending upon your data set, you have to choose whether you will do maximum parsimony method, maximum likelihood method, or this neighborhood joining method. Not that I have seen any tree, neighborhood joining tree you made with the different strains of a fungus or virus or something, but you are doing a neighborhood joining method. What for? If you are doing the strain of the same thing, you should go for the parsimony, not for the neighborhood joining. Clear? So, depending upon your means query, you have to choose what type of tree you want to generate, not that anything you will software, you will tell software will generate, any tree software will generate, but you cannot after that you cannot infer that what actually you would like to say. So, this bioinformatics and other things has many limitations also, there are many good thing, but limitation is that it can only predict, it cannot tell you, it cannot dictate you. Many times it gives erratic result, and most of the times this is due to the user who is using the software, and different subjects are interlinked. So, understanding is very difficult, and it may be possible to make the relationship or models, but in reality they may not work, because we are trying to take challenge with the mother nature, who takes billions of years to evolve us, and we are trying to just sitting on the computer, we are trying to understand what is the relationship, what is the model and all these things. So, we do not know who will tell the last answer, our beloved mother nature or the silicon chips. Thank you for your kind attention. Now, question, anything?
Now the talk is open for any discussion, questions, queries. Of course, you will be handling one more practical. You can reserve those questions for practical because we are running out of time now. Right.
We can discuss. I am here only, so no problem. You can come anytime for your practical analysis also. If you need any help or anything, so you can come.
Bring some set of data for analysis. Whatever you told, character based, distance based, a set of data will give for all the groups. Let them do the analysis using different programs. Okay. Now we'll close this particular talk, and I request Dr. Annapurna to propose the formal vote.
Oh, thank you.
It's a great talk. Yeah.
Yeah. I think you will all agree that it was a very scintillating talk, I would say. And I'm sure most of you will be benefited by this talk. And many of your small doubts or queries, and probably you will now, in retrospect, if you think about this talk and think what you have been doing all along for your sequence analysis, you would know what were the common mistakes that you probably have done. And I hope you will, like he said, after this training program is over, we would at least expect this group of students not to repeat those mistakes. And we also hope that in future, since he is a part of IRA and all of you are students of IRA, if you have any doubt on this particular aspect or maybe others also, I think Anirban, you are available for the student community. And I know he is an excellent teacher. This talk of almost 1.5 or 1 hour 45 minutes is an ample demonstration of that. And please take advantage of this. And like he says, no more confusions on this particular aspect at least. I would profusely want to thank you, Anirban. He is much younger to me, but he has really risen, and I feel very glad when I see such kind of students who have come up from here and are delivering such kind of talks where students are benefited. The whole effort of this training program is to see that the students of IRA are benefited on different aspects of genomics, and I really know that there is a huge confusion as far as genomics and the advanced tools and the advanced techniques are concerned. So our attempt is only to see that that kind of a confusion is removed, and for that please enjoy all the talks in this training program. And in future also, please feel free to come and interact with all the faculty who will be now coming up one after the other that lectures have. Thank you very much. Thank you very much, Anirban.
When I was student, I faced these problems and I developed those things that how really to do, and I am very interested to tell you that how you have come up with those things, and you should do those things, because I also faced this problem because I was an alumni from this institute.
Yes.
So
in fact, we are all alumni of this institute, so that the reason for us to feel this is much more. And this is being telecasted, webcasted to others, and we have sent this link to many of the universities. So I hope the student community in total in India is being benefited by these kind of this kind of talks and other practicals. Thank you very much.
So thank you, Anirban, for a wonderful talk. And with that, we conclude the first lecture for the day. Now we have the second lecture lined up. I'll request Dr. Subramaniam to introduce the speaker.
Good morning to all. I welcome Dr. Kishore Gaikwad, Principal Scientist from NAHEP Plant Biotechnology. We have with us an eminent speaker on the genomics. We have with us principal scientist from NRC Plant Biotechnology. His specialization is molecular biology and plant biotechnology. He has his expertise on the structural genomics and the transcriptomics of the mandated crops like PGNP and wheat. He has a rich experience of handling these crops, especially his focus is on the genomics part of the male sterility in these crops, and also he is pursuing with epigenetic investigations on this. So we will have a talk from Dr. Kishore Gaikwad on genomics. Welcome, Dr. Kishore Gaikwad. Thank you.
So before we start the camera, I just wanted to ask you, there is a very typical talk inside this. It's going to be lunchtime soon, or we should have a small interaction on genomics or bioinformatics or the strategies of sequencing genome, post sequencing analysis and all. If you tell me, I will go according to that. This becomes a very academic talk. What kind of interaction do you want? I do a lecture and go away, or you want to interact with me on various topics? Interaction. All of you? You can have both. Briefly, I will finish my talk and then we will come back to the practical part of doing genomics across all organisms. किधर ठीक है ये काम कर रहा है This one?
This one.
Okay.
ये point है. This is the one.
Yeah, this.
No, this one, sir. Which one is the panel?
This
one. This is
No, this is not. It's one.
Put slightly.
Now it will come. Now this is open.
It is recorder, not mic. So,
Oh, okay.
So because of the camera.
Okay. How many of you have done my course Genomics? Only few? Good. working? Okay. So, good afternoon to all of you. So, what I will do is, what I am going to do is to give you a brief overview on the different sequencing approaches, the different chemistries that we have in the market today, and how one has to design an experiment based on the available chemistries, technology and the databases and so on and so forth. So, very briefly I will take you starting from this history of DNA sequencing, and finally we will come back to the practicality of doing genome sequencing. Next. So, if you look at the slide starting from Sanger sequencing, in fact sequencing was first demonstrated in 1967 by Yu et al. When they sequenced a tRNA earlier in the earlier part of that decade. But the actual sequencing was only demonstrated by Frederick Sanger, and later on there is one more technique, but which got disrupted. Okay. So, if you look at the year 1977, this is when Sanger demonstrated sequencing as a very valuable tool in molecular biology, which can be used. It is still being used as a popular method of analyzing your genomes. Now, if you look at the year 2005, you will see this is where actual genomics started. Although we had lot of genomes published by the end of the millennium, by the 2001, 2002, the major thrust in genomics came only in the year or after 2005. This is when we had the first next generation sequencers, and subsequently in the next 7 years we had a whole umbrella of sequencers of different categories, different technologies, different concepts, each one which own strength and weaknesses, and each one with a different mandate. So, I will take you around all these 5 or 6 because these are very important from a theoretical and practical point of view, and how we can utilize them for doing our day to day genome analysis. So, when you correlate that year 2005, you will see the curves. You will see how the curves are rising only after 2005s. So, what we see now is this is not a very latest graph. What we see now is it's not working. Okay. If you see a growth, the growth is more in lower organisms, lower eukaryotes or prokaryotes. Why? Because they are smaller, simpler and easy to assemble, very basic. The eukaryotes plants will not talk those, they are very complex, they are very big in size, and due to various factors like polyploidy and so on, they become very difficult to handle, because the crowd is related to the smaller genomes. We will talk of the smaller genomes. Now, before that I wanted to know, what do you understand by a genome? Anybody? What does genome ring to you? What does it tell you? One by one, please. That's wrong. Anybody else would like to correct him? Say it again. What haploid content? Chromosomes? So, the simple definition in one line textbook definition is the total genome or total DNA in a haploid cell. She is right. The total DNA in a haploid cell constitutes a genome. So, when people talk of chromosomes and all those things, they are slightly outdated definitions. We talk of haploid cell DNA, very simple, 4 words. So, that is a genome. Now, the second part is, what is genomics? Study of a genome, study of genome. Is it genomics? It is partly correct, but then when you include your proteins, RNA, your ions, your metabolites, that constitute the whole subject of genomics. So, genomics constitutes all that is happening inside a cell apart from the DNA, and when you superimpose all the genomic information with the transcriptome, proteome, metabolome, ionome, that will give you a proper functional pathway analysis for a particular trait or phenotype. So, that constitutes genome and genomics. Now, let us take a look at the genome size, the genome size that we see on this particular planet. We have a very wide range of genome sizes across species, across families, across kingdoms, across genera, and why it is important for a genomic student. We will discuss that point after this particular slide. So, anybody, what is the smallest genome? It has to be a virus, followed by a bacteria maybe, and then your fungal organisms or protozoans, they come into the picture. Now, the largest known genome is anybody largest known genome? So, we will see what is the magnitude of these genomes. So, virus is maybe a 4 to 6 kb in size, is not it? How much is a bacteria? E. coli is 4.5 million bases, 4.5 MB. Do we have a blackboard here? No. How big is a nematode or a yeast? A fungal genome is around 30-40 MB, yeast is smaller in size, nematode is bigger in size. So, as you go across, as you rise in complexity, the genome size increases, although it has no relationship to the organism size and function and so on, or the phenotype, but the genome size keeps on increasing. Although a plant may be smaller than an elephant, it still carries more genome. So, the amount of DNA that you carry has no relation to the structure and the function of that particular organism. So, the largest known genome on this planet is a plant called as Paris japonica, the largest known genome, and that size is around 140 GB. The largest known is a plant Paris japonica. It is a octaploid and the size is around 140 GB. Can you tell me how many MBs? Can you calculate? Put zeros. How much is 1 million bases? How many KBs? How many KBs? 1000 KB is equal to 1 million bases. 1000 MB is equal to 1 GB. So, you can calculate or you can imagine the kind of DNA that we have to sequence. So, if I have to sequence a virus or a bacteria or a protozoa or a nematode, and I have to sequence that plant which is 140 GB in size, I have to use different experimental approaches and different chemistries. So, this is where our knowledge of sequencing chemistries comes into play. Okay. So, there are two approaches of sequencing a genome, two major approaches which have been tried and tested in the human genome, the rice genome, all the major genomes, not in the smaller genomes. Okay. One is called as a whole genome shotgun approaches. I will give you the slides, no need to draw and write all those things. So, one approach is called as a shotgun approach or a whole genome sequencing approach, and the second is called as a bag by bag or a hierarchical approach. So, we will not take, we will come to that once I finish all my sequencing chemistries. The whole genome shotgun is the one which we use for smaller genomes. That is one approach we use for very small genomes. I am not talking of 1 GB, 2 GB, but smaller genomes we take up the whole genome or WGS approach. So, these are the chemistries that we have in the market now. These are 7 to 8 chemistries that we have in the market. A couple of them have been stopped or outdated or no longer in operations, but we have to see how they worked, because that was a wonderful principle upon which they operated, and they were really good as long as they were there in the market. But these 8 or 9 sequencers are the only ones that we have as a tool for sequencing. I think I can skip this one. Everybody knows? Sure? No? I need a blackboard actually. Anyway, we'll wait till the blackboard comes. Can you see the sequence? Can you read the sequence for me? So, that is a strand I want to sequence. That is a fragment of DNA I want to sequence. So, what do? I take 4 tubes. And in each tube I put a mixture of DNTPs, normal DNTPs, DNA polymerase, sequencing primer, and only one dideoxy DNTP. The role of a dideoxy DNTP is to terminate or block the growth of a DNA chain. So, in my first tube I have taken dideoxy ATP. In my first tube I have a dideoxy ATP. So, if you can write the sequence and if you can do it by your own, you will understand it much better. So, if I take dideoxy ATP, if I take the first tube, if I synthesize using primers and DNA polymerase, what do I see? How many fragments can I get theoretically? You write and then you calculate. So, you stop at T, wherever you have T you stop there. How many T's you have? 4. So, you will stop at 4 places. So, that means you will get 4 fragments, and the size will be depending on the position of the T in that particular fragment. So, the position is at 3, 6, 7 and 10. So, that would be the size of your synthesized or amplified or sequencing products. Got it? No? Clear? Similarly, the tube 2 where you have only one DNTP dideoxy G followed by C and followed by T. So, now you have a mixture of products in tube 1, tube 2, tube 3 and tube 4. Then you run this on either a capillary or a sequencing gel, and based on their size, because these are all labeled by fluorescent dyes, based on their size you can calculate which is the first base, which is the second base, which comes on the right panel. So, this is a basic Sanger sequencing technique. Should I repeat? Unless you do it on paper, you won't get it. Is clear? Now, Sanger sequencing can take you up to 1 kb, that is a maximum read length. Okay. So, Sanger sequencing can take you up to 1 kb, that is a maximum output for a single fragment, but if you can run multiple samples, you can finish 384 samples in 2.5 hours. So, your output would be 384 into how much? 1 kb. So, each run would give you 384 kb. You can note down, because that is important. As we go ahead, we increase our data exponentially. Is this part clear? Next. This was called as a first generation sequencer. This is based on the principle of pyrosequencing. There is no radioactivity, there is no fluorescence. It is totally based on the luciferase activity or normal light. So, that is why the name pyrosequencing. Now, the concept is very simple. Isolate your DNA, break into smaller pieces, attach to beads. Now, this attachment is something that we do not control. It is all controlled by the solution, which is not known to us how they do it, but in the end you get one fragment bound to one bead. This is how it operates. And then a PCR is done, because these parts are known to us, the adapters are known to us, and from one fragment on each bead you make millions of fragments. So, this is called as clonal amplification. So, one fragment becomes many fragments and it gets attached to the same bead. So, one bead has, so one fragment made many copies, they are all attached to the same bead. So, likewise you generate millions and millions of beads, each carrying the entire DNA or genome of an organism. Next. Then these beads are plated on a small chip. It is called as a picotiter plate, PTP, and then your enzymes are added, luciferase, sulfurylase and polymerase, and by the time the bases are added, this chain will grow like this. Each base is added sequentially. Remember, not all 4 bases are added at the same time. One base is added. Wherever the base gets attached, light will be given. So, you know, the computer knows that this A has been added. Wash, add the second base G, look for light, add the third base C, T, and keep on doing it. So, sequentially one after another the bases are added and the light is recorded and the sequencing grows. So, this is based on pyrosequencing, and the maximum read length is 1 kb, again for as same as Sanger, but the data output is close to 1 GB. Data output is close to 1 GB. Next. Next is a sequencing called as a Illumina sequencing technology. It is still a very valid technology. We all use Illumina. You all will use today or tomorrow. It remains a very strong technology. Nobody is able to beat it. It remains the king of sequencing as far as whole genomes are concerned. So, the concept here is same as we saw in the pyrosequencing. So, you fragment your DNA, you add adapters, and then this DNA which is bound to adapters is attached to a glass slide called as a flow cell. It is a very small glass slide called as a flow cell. These fragments are then made to fall back they fall back and they join a second adapter on the flow cell itself. These fragments of DNA, they fall back, bind to a adapter which is already there on the flow cell, and that makes a U structure. So, all the fragments will get converted into these bridges, and then a PCR is conducted. This PCR will convert this single stranded to double stranded at all the places and keep on doing it into multiple copies, and this process is called as bridge amplification. This process is called as bridge amplification. So, from 3 copies now you have maybe 3 million copies. So, likewise the whole genome gets amplified millions of times. So, the starting material is very, very less. You can work with nanograms of DNA. That is the best part of this particular chemistry. Now, once PCR is completed, you have something like this. So, all these fragments are copy of this one fragment. So, this is called as a cluster. So, one fragment makes one cluster. Likewise, there are millions of fragments. You get billions of clusters on a small glass slide. Then this particular glass slide or flow cell goes for sequencing. So, this is PCR based amplification. Starting material is very less. Ultimately, we need to sequence it. So, next slide. So, sequencing goes like this. So, all the 4 bases are added, they are all labeled with the fluorescent dyes, all are with different colors, red, blue, green, yellow, whatever is available. And then as one base is added, suppose a T is required here on this particular cluster, it has a blocking dye called this red one, the pink dye. The moment T is added by DNA polymerase, after that no more base extension takes place, same as dideoxy. One base added, stop, cannot grow as a chain. So, moment it stops, a second step is conducted to remove the blocking reagent. The more bases are added. So, this cycle keeps on continuing, add a base, block it, remove the block, add the second base and keep on doing it. By doing this, you can expand the chains at the same time, although this process takes a week. This sequencing runs for at least 7 days, 5 to 7 days, depending on how you want to run the machines. So, this chemistry can generate up to 2 TB data. This chemistry can generate up to, there is a system called as a NovaSeq, Illumina NovaSeq. It can give you 2 TB data. How much is that? 2 TB in terms of GB? 2000 GB. How many genome sequence in one run? How many human beings one can sequence in one NovaSeq run? What is the size of human being DNA? 3 GB. How many human beings you can sequence here? So, 2000 divided by 3, more than 600. So, this is the power of Illumina. You cannot do this on Sanger, you cannot do that on pyro sequencing and the ones I am going to describe. So, that is why this machine remains one of the best we have in the current generation of sequencers. Next. In between we have this system called as a ion torrent technology. As the name says, ion, it is based on production of ions, especially hydrogen ions. It is based on a physics transistor technology. The concept is very simple. You fragment DNA, attach to beads and attach this to a voltage detector. Attach the chip to a voltage detector. You add one base, suppose I add A. Wherever A is incorporated during sequencing, it would see a change in the pH, because your hydrogen ion is released. So, that means one base has been added. So, no radioactivity, no fluorescence, no pyrosequencing. So, very cheap platform, very simple to operate, and very robust as long as it lasted. Some people still use it, but it remains generally out of the market. So, this sequencing is based on the change in the voltage gradient, and you have to add each base sequentially. You can't add all the 4 bases, because they are not labeled separately. That is why the name ion torrent. Next. Then sometime in 2008, we had a new term in the market called as a single molecule sequencing, and the company was Helicos, or the product was Helicos. The principle was very simple. You again generate fragments of DNA attached to a glass slide, and then you can extend using polymerase enzyme the chain of DNA which you want to sequence. So, the genome can be sequenced again on a small glass slide. The length in this particular chemistry was only 50 base pair. You cannot generate more than 50 base pair. Unfortunately, this chemistry is now outdated. They don't provide any support. They don't use it. So, we will not discuss more about this particular chemistry. Next. So, the real single molecule real time sequencing, SMRT as it is known, is called as a PacBio, chemistry. Now, PacBio runs on the simple operation that if you take a small glass slide with a small nanopore or a micropore, at the base of this micropore there is an enzyme called as a DNA polymerase. They call this some other thing. DNA fragments, long DNA fragments are passed through this channel. So, the DNA enters here, and the 4 bases are labeled here. As a base is added, a color is generated. You can record the color. Sequencing has been completed, and the chain moves out. So, the chain moves in as a single stranded, it comes out double stranded, and that double stranded data is recorded in terms of light. So, 4 types of light will be generated, because 4 dyes are there, and you can generate really very long sequences. So, anybody who wants to do long sequencing, this is a chemistry you can use, PacBio chemistry. Next. The last announcement in sequencing came in 2012 by a technique called as a Oxford nanopore sequencing. Metagenome, then again, then in that case you need to see the cost. So, PacBio is expensive, Illumina is cheaper. Which one? Error rate PacBio is slightly lower than Illumina. If it's
single-stranded RNA virus of 8 kb, how should I proceed? Like
from
whose reference genomes are available?
if it is not available, then it becomes a de novo assembly.
Like reference-based assembly I should go for, but how should I proceed for sequencing? Like from a plant?
From a plant, can you amplify it? Can amplify?
Any sequences
Can you amplify the whole virus?
yeah, I can match.
If you can amplify, then Sanger remains a better choice, because nanopore and PacBio will be expensive.
What is the difference between draft genome sequence and whole genome sequence?
Draft genome is something which covers 70%. If your genome size is 100 MB and you have sequenced up to 70 MB, that remains a draft genome.
They are taking the data from the draft genome.
Yeah, it is draft genome data will be always poor. You won't get good quality data. Draft genomes.
Sometimes we get RNA, we want RNA sequencing. So for that, which platform will be suitable?
Which organism?
So it's means crop.
It's a plant, and depending on how many samples you want to run,
So
6 samples. So Illumina remains, it is much cheaper. If you do one sample, if you put in that 2 TB, you can put one sample or you can put 1000 samples. So one sample cost would be same. If you put 1000 samples, it will divide it by that much. So that remains a better technique. If you multiplex, you can multiplex many genomes at the same time and put into the same lane of that 2 TB machine. You can get multiple genomes in 3 to 5 days.
so means cost for one sample is similar to the.
No. You see, all this is, suppose you talk of Illumina, there are 8 lanes in a high-seq machine. So in each lane, you can put around easily transcriptome 16 samples ideally. So if you put one sample, the cost would be high. Put 16 samples, cost would be less. Of course, your data gets distributed per sample. That's what, if you can amplify it, then you can, then Sanger remains a cheaper. You can use all of these chemistries, but Sanger remains a cheaper ones. Although Sanger takes time. Some people, what they do is they take the virus infected plant and they sequence the entire plant. Some people do that. So for that you can use Illumina machine. Yeah, you can do that. You can do that and then separate them by bioinformatics. You can separate them. How many pools? No. You have to cut it into smaller pieces and then go for Sanger sequencing. If it is very small, 8 to 10 kb, then it's a very small size. Better to cut it into 3-4 pieces and then sequence it, so that you get more depth, more coverage.
Sir, in case of Sanger sequencing, whether single 1x is sufficient or we need to go for 2, 3, 4, 5?
Sanger 1x will not give you good data. If you are assembling a genome
Like small, like 2 kb or
even if it is 0.5 kb, you get a 10x data.
10x.
It's not acceptable nowadays that you sequence once or twice and then you work upon that. It's not acceptable. 10x is a gold standard for all kinds of sequencing.
For sequencing any like those for going for genome or something, what should be the sequencing method?
Virus,
For 50, I think fungal genome.
so for a fungal genome of 50 MB size or genomes in that size, pick up at least 2 chemistries. you can take Illumina as one, because it will generate lot of data. So, that becomes your backbone, and then you can either pick up nanopore or SMRT or PacBio, and then combine them, which is called as a hybrid assembly. So, when two chemistries come together, they improve your data quality. If you rely on one chemistry, you will have error rates. So, better use 2, 3, 4 whatever you can, but smaller genomes 2 is fine. So, it should be done in a very less cost.
You consider it is at 389 MB, somewhere 400 MB, somewhere 420. So what is the criteria to actual size for this? Like
Actual size is based on two things: fluorescence cytometer. If you do a FACS analysis, it gives you almost 90% range. Sequencing, if you do a proper sequencing like we did in rice or tomato or wheat or pigeon pea, that is also a correct estimation. But I should tell you one thing here: 10 to 15% of all sequences in any genome remains unsequenced. So, if it is 389 MB, you put 10% which we are not able to sequence. All genomes, even human genome, 10% of the genome remains unsequenced. We do not know why. The chemistries are not able to tackle those sequences, or we can't assemble them. So, that part remains out of bounds. So, the size will keep on fluctuating. So, best way is to you pick the size which is generated by the help of physical mapping data. So, if you just take DNA, put into a sequencer, you will get some genome, but that is not a correct way of doing research. The one done in human genome or rice genome, they are the gold standards. So, first you make a genetic map, a physical map, and then you sequence them, and then you make the sequence into linkage groups. That is the data you should use.
Any more questions from anybody?
Price I will tell you after this, once this is over. If you want to know the price, once this is over, I will tell you the price. I am not supposed to talk the price online. They would have some questions, I think, during their, maybe after this. They would be handling it.
A very nice talk about different sequencing technologies, starting from very basic samples and right up to SMRT sequencing technology, that is the PacBio technology. Dr. Kishore is very much with us all the time. He is always ready for any kind of technical support, clarifications on your sequencing queries. You can meet him. He is sitting in
Contact details if you are not taking.
the phone number is already there.
Anytime you want to design an experiment, please contact any one of us who are into this for a better structured experiment.
Yeah, that is very, very important. So you should know the depth of the well before jumping in, right? So talk to him, have a thorough discussion. We all jump together, and he will offer you a suggestion. What kind of technology you have to use for the kind of genome size. Somebody was asking, I have 8 kb, can I go for NGS? That is not needed. So you can do it with the scientists. So there are criteria you have to really follow before choosing a platform, right? So likewise for any sequencing technology, it is driven by the sample what you are handling. So keep Dr. Kishore in your research team, try to take his expertise and consult with him and I'm sure that will put your project in the fast track mode.
I'll give my slides for you.
Exactly. So with that, we'll conclude this forenoon 2 lectures. And I take this opportunity to thank Dr. Kishore. You can give it. You can. Photos. So, Dr. Kishore is one among us. We have a joint project funded by ICR that is called as CRP Genomics. SAR is handling huge genomes. We are handling small genomes. So I requested him to deliver a talk on small genomes. So that's the reason why he narrated the basics of sequencing technologies which is useful for small genomes. Thank you, Kishore, for your valuable time. I'm sure the students gained a lot. They will continue to keep in touch with you, and thank you once again. Now we will break, and you can. The lecture session is over now, and afternoon by 2 o'clock, let us assemble back here, and we'll take stock of what we need to do in the afternoon practicals. So we'll continue the practical what we started yesterday, and we'll go for new practical today. It is PCR technique. And the team has kept everything ready in the lab. So you set up PCR and you can leave today's practical. We'll continue the today's practical, the PCR, tomorrow only. Right? Now we'll take a break and have a quick lunch and we'll come back by 1:45, not 2 o'clock. Right? Try 2 o'clock sharp, we'll start the practical. Thank you so much.